So since this course deals with experimental approaches to study and understand the function and plasticity of cells, I actually think that our new experimental approach to study programming and reprogramming in these cerebral organoids may fit very well. So maybe we can do it a bit darker because there are some pictures in the front. So far, mostly, and except from the end of Paula's talk, we heard about the endogenous plasticity of cells. And so now I want to make you consider the programming and reprogramming of human neurons rather as a forced plasticity, meaning that we change the identity actively of the cell. We change the identity by imposing or overexpressing some transcription factors that are important also during development. So, as also mentioned briefly, whereas embryonic stem cells and pluripotent stem cells are pluripotent, so they can generate cells from all three germ layers, direct reprogramming takes a shortcut. That means by actively overexpressing transcription factors, we force the cell and change their identity. So by now, quite a few groups have shown direct reprogramming also into induced neurons. And of course, the studies that have used fibroblast as starting material offered very important insights into the process of reprogramming. I think that cells endogenous to the tissue where the degeneration takes place offer very promising therapeutic insights. For sure, we should mention here astrocytes and NG2 glia, and also Benedikt has shown that, but also parasites that were there, and Paula nicely included them in this scheme here, can be reprogrammed into induced neurons, and hopefully by the end of the talk I can convince you that this is indeed a very interesting cell population that should be considered as a cellular and therapeutic target. So what are parasites? In general, very quickly, ubiquitously present in the entire body, usually associated with endothelial cells, and here you can schematically see that they are enwrapping endothelial cells. They come in different flavors, and a fact that is very neglected so far, and I think very important, is the fact that these parasites do exhibit endogenous plasticity. So also parasites, or Giulio Cossu could show that in the mesoderm, parasites in the skeletal muscle can be activated following injury, can be recruited, and then also actually contribute to tissue regeneration. More importantly, in the CNS, Christian Göritz and colleagues have shown that in the spinal cord there's a specific subpopulation of parasites called type A parasites that, following spinal cord lesion, start to proliferate, they detach from their endothelial counterparts and then actually form the scar core. And more importantly for us, when we then move to the human system, we could show that human parasites can be isolated from the human cerebral cortex and then can be reprogrammed. And this I want to summarize in the few next slides. And I should mention that we were very lucky to establish a collaboration with the neurosurgical department in Munich. So now on a regular basis we get specimens from human patients that undergo neurosurgical interventions, and this is cryosections of these brain samples. You can see the parasite labeled in red sitting on top of the endothelial cells. These are a few markers, and as mentioned just right now by Paula, for parasites it's very similar. There's not a unique marker that labels parasites. It's a combinatorial expression of markers and genetic profiling that you can do to then conclude that you work with parasites in vitro. So then what we did, and this is how a typical parasite culture looks like, is rather heterogeneous in terms of these markers, PDGF receptor beta and SMA. When we have this culture, when we have these cells and we use retroviruses and overexpress two transcription factors, namely ASCL1 and SOX2, we can convert, and this is a fairly simple protocol, so we transduce the cells, put them in low oxygen condition in differentiation medium, and then after 3 to 4 weeks we can see, and you can appreciate, there's a dramatic change in morphology, acquisition of a neuronal phenotype, including the expression of neuronal markers like beta-3 tubulin. The efficiency is pretty high when we now switch to this polycistronic construct. 50 to 60% of all the cells that do overexpress MESH1 and SOX2, ASCL1 and SOX2, do convert to neurons. And very importantly also, we were able to show that these cells, once converted, do also exhibit some electrophysiological features of bona fide neurons, even though immature. So the next step now, no matter where we go, is always to see whether these cells are actually really, truly relevant, whether they do have a true relevance, and no matter what we do, we will always get to the point where we have to see, in other words, how do we study functional properties of cells? How do we study the integration of these human in vitro reprogrammed neurons into pre-existing neural circuits? Even though there have been studies shown that in vivo, and also by Benedikt, you can directly convert astroglia or glial cells into neurons. Of course, this is out of debate in the human system, and you can do then transplantation of these cells in a mouse model. However, also this has some limitations. So I think that the seminal finding of organoids has really opened new avenues to study the functional relevance of in vitro reprogrammed human neurons within a system. This is completely composed of human cells. So I will show you our organoids that we also, similarly to the protocol that Paula uses, follow mostly this protocol by Lancaster and Al from the Knoblich lab, and this is Radhika Menon, our PhD student. And so this is pretty much schematically shown, but just to give you an idea, so this organoid is pretty much via an embryonic body state. These cells form, without the use of any patterning and growth factors, discrete brain regions, and Paula also showed you, but I want to show you another example here, of SOX2 positive cells. So these germinative zones are formed of SOX2 positive cells surrounded by beta-3 tubulin cells. We also have these apical or radial glia-like cells that are at the apical surface of these ventricular structures. We do have TH positive neurons, rather in older organoids, also some reelin positive Cajal-Retius cells. And very interesting for us, if we wait long enough, we also see, maybe not, we can see a lot of interneuron subtypes, again also just based on morphology, as mentioned before now many times. But we can see calretinin positive, calbindin, somatostatin, and also GABA positive or immunoreactive cells. Let's say this very careful. So what is also very interesting now, and the next question for us is to see what happens if we don't leave it up to the pluripotency and the default differentiation pathway of these cells. What happens if we interfere with this, and can we change the cellular composition of these organoids, and with that also then maybe influence the functional integration of our transplanted cells. So what we do, and luckily there was introduction enough, so I think I don't need to go into detail. During development, sonic hedgehog signaling pathway plays a very central role in the ventral telencephalon, where in the ganglionic eminences interneurons are formed, and we heard also about these transcription factors that play an important role there. So when we looked at our organoids, we found that in approximately 1/3 of all the organoids, we have NKX2.1 positive cells, so ventral telesymphatic progenitors. But when we added pormorphinemin, which is a sonic hedgehog agonist, we actually increased a lot the amount of these and the occurrence of these NKX2.1 positive progenitors. And right now we are characterizing in detail how this increased occurrence of NKX2.1 positive progenitors influences also later on the interneurons generated within these organoids. And why are we doing this? We do this because we want to assess whether, once transplanted our induced reprogrammed cells, actually the cellular composition of the organoid would maybe make a difference in these transplanted cells. So let me show you this, our experimental approach. Again, it's a bit light, but maybe you can still see it. And I want to also introduce a term that Radhika came up. So these organoids are very interesting for lots of different reasons, to study development, neuronal migration. But what I want to focus on really is to use them as so-called brain incubators. So This is a system composed fully of human cells, and we want to study functional relevance of our human reprogrammed cells. So we thought it would be interesting to see what happens if we transplant the parasites. So we transduce them with the retrovirus ASCL1, SOX2, which also has GFP. We sort the cells. We have made sure that they, as you said also, Paula, are not affected by the sorting. They are still very alive and can still be reprogrammed. Then we transplant them into these organoids. And then we wanted to see whether we can increase the maturation, can maybe also live image this conversion process, and whether we can promote subtype specification, and finally also analyze integration into these circuits. So let me show you our first examples. So this is parasites undergoing reprogramming, transplanted into the organoids. Then we do cryosections, 40 micrometers, and then we check the cells. So you see they're still there, but more importantly, the green cells, which are the parasites within the organoids, do also exhibit some induction, or they show signs of beginning pluripotent reprogramming, because they start to upregulate beta-3 tubulin. Then you can see another example here. I think it's very evident that these cells are able to be reprogrammed within the organoids. They acquire nice morphology of neurons, induction of beta-3 tubulin expression, and now we moved on to always co-transplant, control transduced, and ASCL1 and SOX2 transduced parasites, and I think the direct comparison makes it clear that we can see and we can assess reprogramming within these organoids. More importantly, though, and again I want to show you, this is all very early days, I think, from the organoids, and we need to establish a lot of things. Also the dimensions of them, they grow up to 3 to 4 millimeters. So if you want to monitor an image and visualize reprogramming, you have to find ways how to visualize this. So we adapted the clearing protocol, and I want to show you here that we were able, after clearing, within 400 micrometers to see actually and monitor or visualize the transplanted cells. This is the control transduced cells, and I think in the direct comparison you can also see that the cells undergoing reprogramming have very evident morphological and apparent differences compared to these control transduced cells. And finally, and this is also just to give you more an idea of how we think we can use them as brain incubators. I want to show you that we were also able to do some sort of short but still live imaging following transplantation. So this is again apparent differences between control and mesh1 SOX2 transduced cells. You can see that we could image within 20, this was with 2-photon microscopy, within 20 hours we could see the dynamics of these cells, and indeed I think it's very interesting and insightful to study also the dynamics and kinetics of reprogramming. And we were now, and we will follow up on this more, let me focus your attention on this cell. We were hoping, and we hope we will manage also, to then be able at some point to really witness reprogramming in life. So this cell, which looked different before, now exhibits a few neuronal processes, and this is just to show you what we want to use this for. So to conclude, I think that cerebral organoids, apart from many other things, can really serve as a suitable tool to study reprogramming within a human tissue-like context. We can also manipulate cerebral organoids and then see whether the cellular composition would actually have an influence of the reprogramming in the organoids. Then we transplanted these parasites, and I think this will be a useful tool for us. And finally, of course, we tried our first approaches here also to patch the reprogrammed, not only the endogenous cells from the organoids, but also the transplanted reprogrammed cells within the organoids. And again, I have to say, it's early days. We have to figure out ways to do, let's say, somewhat acute slices of these cells. And with collaboration with Bernd Suter, I'm sure we'll succeed with this. So that's the end. And I want to thank these people and you for your attention, and thank you.
I have a technical question and then one more conceptual. From a technical point of view, when you image these organoids, do you take them out of the spinning bioreactors and let them grow in plates for a certain number of hours? And if you do that, do you think that they change their morphology? What do you do there exactly?
So we take them out and then we do embed them in agarose, and then we do 300 micrometer slices, and then we right away, I mean, we have to equilibrate a bit, but after an hour we take them to the microscope.
And then how long can you image for?
So the first, I mean, the movies here were like 20 hours. We did 4 days also, but we were not lucky to find the right spot. The problem was after 4 days we had a contamination. So I don't think, and they collapse a bit.
Like every slice.
But,
right?
And especially when you want to study human reprogramming, of course, this will take more than only 20 hours.
Yes.
But I think that right now 2 or 3 days is feasible. So then we'll have to.
Okay, and then the conceptual one is, so these organoids, like every basically neuron that is ever made from an iPS cell are young, right? So they are basically a replica of a early fetal human brain, let's say. Do you think that that matters in terms of environment to allowing these parasites to reprogram, that you're testing basically the effect of a fetal environment on the capacity of the cells? Is that an advantage, or what do you think about it? Just more conceptualism, almost like philosophy of.
I think that in general it may not, it may not be a huge problem if you have rather young cells, because it's very neurogenic. However, it has limitations, because also these young neurons, as you mentioned, have other functions and characteristics than older ones. Maybe one could consider to transplant older neurons derived from iPS cells, even though the question is how
old are they really old?
So I think it's very neurogenic, but I think it's just a limitation of the system.
Thank you.